So when I was asked to give this talk, I was trying to think, and how in the world do I connect my chemical engineering education and all those technical roles to what I'm doing now as the program lead for COVID-19 vaccines at Moderna. And when I thought about it, I thought whether you're in the lab, whether you're in the pilot plant, or whether you're figuring out what kind of booster do you need for the Omicron wave, it's really all problem solving through science. So training in engineering, breaking down problems, figuring out how to solve them, it serves you well in all of those types of roles. So I want to start a little bit about how I got to where I am today. So I thought I'd start at the beginning. I was born in Charleston, West Virginia, so over the mountains. In the other direction, the far west of her map. I found out early that I like math and science, and I was pretty good at it. And this is really the only online record of my existence. So it was kind of comfortable that this is all you can find. So you may not be able to figure out which one is me. That's me in the first place row, 8th grader, with my teammates Chip and Alex. We swept the county math field day that year, and I went on to actually win the state math field day in 7th and 8th grade. So when it came to what did I want to do in my career, growing up in Charleston, West Virginia, chemical engineering or the chemical industry was a big part of the economy. And it was also a big part of my family economy because my dad was a chemical engineer as well. So it was just something that kind of presented itself to you if you were good in math and science. What about chemical engineering? But I thought about where I wanted to go for college, and I didn't want to go to just an engineering school. I wanted to go to some place where I could learn about a lot of other different things if I could squeeze it in. So that's why I chose UVA. And I did manage to squeeze in a couple of history classes, history of American law and US diplomatic history, Shakespeare classes, modern American authors, and like many people, intro to econ. So both the classes and then having smart people that I met and became my friends who are involved in all different areas, I think was a huge benefit of going here. So I also had a couple of research experiences, one at Dow Chemical and one in the basement of Thornton Hall. And that's why I decided to go on for grad school. And as you can see, I'm continuing this dome theme throughout my life. After MIT, I decided that I really was interested in healthcare. And so I looked at the pharmaceutical industry, and as it turned out, it was kind of the perfect blend of my dad, the chemical engineer, and my mom, my working mom role model, who was a pharmacist for 52 years. So that's how I ended up at Merck for about 20 years. I'm going to spend quite a lot of time talking about my career there. Then I was at Teva for a while doing some cool work on drug device combinations. So I looked at an implantable drug delivery device. I also looked at I was also a program lead for developing inhalers for people who have very low lung capacity so that a normal inhaler wouldn't work for them. At Takeda, I really learned to work across cultures. Takeda is a Japanese company. I did a lot of travel to Europe and Japan during that time, and also learned about the business of what happens when you combine two companies. So my job there was integrating Shire Pharmaceutical Sciences and Takeda Pharmaceutical Sciences into one overall department. So kind of had finished that up by early 2020, wasn't sure what I was going to do next, and a recruiter called me about a job at Moderna. So that's how I ended up there. I'm going to spend most of my talk talking about sort of, I guess, a little bit about graduate school, a fair amount about Merck and my twists and turns through Merck and how that ended up leading to my work at Moderna. And I'll save some time at the end to talk about what I actually do there and save even more time for questions. So also trying to lace a few lessons in there. So I think learning number one is that in every job or every experience, you're going to learn something useful, but it may take a while to figure out what. So in grad school, I found it was kind of like nested problems, almost like those dolls where you take apart and there's another problem and another problem. So my big picture technology goal in grad school was around separation. So how do you optimize purification by liquid-liquid extraction, which is a very common purification technique where a solute is taken from, say, an oil phase into a water phase, and that ends up purifying it, right? So then the next question that I was specifically trying to answer was how does surfactants, which are molecules that are polar on one side and soluble in oil and soluble in water, and nonpolar on the other, soluble in oil, line up at the interface. How does that affect the rate of solute transfer from one phase to the other? The answer to that, I guess the biggest question was how do you actually measure that transport across the interface? And the answer was lasers, mirrors, and video. So I'm not going to explain the whole laser mirror setup. It's been 30 years, not going there. But I guess the one thing is that, I'm not sure how to best show this. Oops, that is not what I meant to do. Let's go back. The test cell, which is in sort of the upper side in the middle, is where the laser needs to go through my interface so I can measure on either side of it. Problem is, you've got this meniscus, which is in exactly the place that you want to be measuring, so the laser reflects off and it doesn't work at all. So the next problem was how in the world do I make a measurement cell so that I have no meniscus, I have a perfectly straight interface? I had absolutely no idea. So I spent lots of time talking to my friends, complaining about this intractable problem, trying to figure it out, until my friend Mark Allen, who is now a professor at Penn, but at that time was a chemie student working in the electrical engineering department, he said, hey, you can treat the glass very precisely so that at the bottom of the line, though, there's low surface tension with water. At the top of the line, there's low surface tension with oil, so that you get a perfectly flat meniscus. Like, you can? How do you do that? So he got me introduced to the electrical engineering department. I got trained in photolithography, and I was able to make the perfect measurement cell to do my experiments. All right. So 4 years of doing this experiment, doing this thesis project, what did I learn? Well, yeah, I learned about extraction. I learned about physicochemical hydrodynamics and surfactants and lasers and mirrors and photolithography. But I think what I really learned was what it takes to be a successful engineer. So it takes curiosity, and not just curiosity about your field, but curiosity about all the different nested steps in somebody else's field and lots of things. I knew it took independence. I mean, it's your project, so no one's going to tell you how to do it. You have to figure it out. There's a lot of sort of taking it on yourself to be successful. Resilience, persistence, all those synonyms, you're going to fail a lot, and you need to pick yourself up and just keep on going. So there's a lot of resilience. At Moderna, we call it being relentless. And then what surprised me was really that I couldn't do it without collaboration, because I knew all about the independence part, but it was really the being able to collaborate to get ideas from other people that was perhaps the most important thing that I learned. So I mentioned that after grad school, I wanted to work in healthcare, and I went into a department called Chemical Engineering R&D, so clearly very technical, very related to what I'd learned. And I was developing processes to make drugs to treat diseases. So I worked on an asthma drug, I worked on migraines. And my role is to take the chemical synthesis and the separation process that a chemist had developed in the lab hood with flasks and beakers, and figure out how we could make it at large scale to make material for clinical trials and eventually have a manufacturing process where you could make drugs to distribute and sell. So this is just a tiny little, like my first month on the job thing that I did, but the chemist gives me the process and they're telling me about this chlorination reaction. And what they do is pour a little bit of the reagent into the beaker. This is actually the same thing you use to chlorinate your pool. And then you're supposed to pour it very, very, very gradually into an Erlenmeyer flask with a stir bar. So you don't pour it so fast that you get 2 chlorines per molecule. You only want to get 1. And they figured this is going to be easy. You just go do the same thing in the pilot plant. And I was like, no, we're not going to take the chlorinating agent, stick it in a bucket, and ask the operator to gradually pour it into the reactor. We're going to dissolve it up in water. We're going to get a pump. We're going to pump at a set rate, and that's how we're going to do it. And they were like, wow, how'd you think of that? I'm like, yeah, I'm an engineer. So, yeah, So that was a simple problem. Oh, yeah, I guess transitioning to this. So after a few years in chemical engineering, I'd learned tons of stuff. I wasn't sure that this was for me. I wanted to think about maybe more broadly, getting involved more broadly in drug development. I started exploring other possibilities. And my boss's boss found out that I was exploring other possibilities and called me into his office for a meeting. And, we talked about why I thought I might want to do something else. And then at one point he looked at me and he said, Anne, you know that no one cares more about your career than you do. I was like, what? Like, what about my manager? Doesn't my manager care the most about my career? Don't you care the most about my career? And it took me a while to realize that that was really good advice, that you actually are responsible for your career and where it's going to go. So with that in mind, as you get to sort of different waypoints along the way, you really need to assess yourself. And what do you specifically, what do you truly do well? What do you like to do? What's enjoyable for you? And how do you think you can make a difference in the world? And I think that can help you guide your decisions. Another thing is you got to let the right people know, because people don't know what you want to do. They don't assume that people recognize that you want that position or that assignment or want to go in a different direction. And the right people could be your boss, they could be somebody else's boss, they could be your friends, but it is important to get that information out there. And then the other part is really say yes to opportunities. And that's even if they do unbalance your life, because work-life balance is important, but it doesn't mean every day of your life is going to be balanced, or every week, or maybe even every month. And I think you need to figure out, at any point in your life, are you ready to take a leap and really get off kilter? And sometimes those leaps and being unbalanced are the times you really grow. Or maybe a smaller pivot where you have sort of one foot firmly planted on the ground and another one pivoting. So, all right, my daughter hates these pictures, but my sport in high school was dance, so I kind of love them. So anyway, so it could be a pivot or it could be a leap in your point shoes. But anyway, either way, change is going to require some movement. So my first pivot was from drugs to vaccines. My husband had gotten a position in Pennsylvania. We were in New Jersey at the time. I needed to leave the chemical engineering department. We actually had an infant son, so it's not like we could really handle living apart by 2 or 3 hours. And I thought about what I liked doing, what I wanted to do going forward. And here's where I were. It was at chemical engineering. It was me, the chemist, and the pilot plant operators. And it was pretty contained group of people that I interacted with. I really wanted to get more of the big picture of how drug development happened. And I knew that if I moved into vaccines, it would be a much broader group. So I'd be leading a team of all of the people developing developing the vaccines, doing the testing, transferring to manufacturing. And then I'd also be on the overall project team so I could understand how clinical development was happening. So that's why I chose to move into that job that was a lot different than what I had been doing before, rather than a smaller pivot to something similar. And my first vaccine project was really great. It was really complicated, and it was complicated for a number of reasons. So this is just a paper that we wrote in the late 90s about a polysaccharide protein vaccine and the purification process. But what led to that was first discoveries in biology. So Streptococcus pneumonia causes a lot of disease in both the elderly and infants. There's a polysaccharide coat around the bacterium that is sort of is what causes the immune response in the body. And it was determined many decades ago that if you isolate and purify that capsular polysaccharide, that chain of sugar molecules, that can be used as a vaccine for adults and protect them from disease. Doesn't work for babies. So children under 2 have an immature immune response and don't react to a polysaccharide vaccine. So if you want to make a vaccine for babies, you need to grow up the polysaccharide and isolate and purify, grow up a protein, isolate that and purify it, and then react them together. And after you react them together, the yield tends to be very low, maybe 10 to 20%. So you have to get rid of all that polysaccharide that didn't react, or it won't work as well. And there are other problems with having a lot of polysaccharides in there that aren't doing anything for the children's vaccine. So that's where the purification came. And we purified by filtration, which is circulating the stream around, and the smaller polysaccharide goes through the filter, and the conjugate stays in until the polysaccharide stops going through the filter because you have a gel layer and it can't go through. So this whole paper was how we pressurized the backside of the membrane to blow that gel layer off, and how we optimized it so that it would work at large scale in manufacturing. And that was it. So started with polysaccharide, add protein, add that all together, and that was our complicated problem that we solved. And it was very exciting, very, very rewarding, and that was my introduction to vaccines. So continuing the path from focus to broad, here I am in vaccines. And then I decided to take a detour. So in 1999, I had my third child, Emily, who's now a grad student. And I found that being in the lab was very challenging. And I was looking for a job where I could still learn, where I could use my skills, but also where I had a little more control over when I did my work and where I did my work. So I took a job in technical writing. And what I was doing was organizing and writing and editing documents for the FDA. So I worked on a pneumonia vaccine, and my biggest accomplishment really was writing or being responsible for the biologics licensing application for Gardasil, which is the vaccine that probably you all know about, but it protects women from cervical cancer and protects people from various other cancers as well. So that job really ticked all those boxes. It allowed me that flexibility. I was learning about regulatory aspects of how you get something through the FDA. But what it didn't give me was much visibility. I was much less visible than when I was in that team leader position. So what I'd say to you is, if you take a job like that and you get a little off the radar, it's really important to find some way to be more visible. It's kind of like when you go on a hike and you're hiking by yourself, you need to tell people where you are in case you get lost so they can find you later. So what I did was I found out that this governance committee, so this group of vice presidents who was responsible for making decisions about how vaccines were developed and what got priority and all of that. So I found out that they needed someone to do the meeting management. So coordinating all the meetings, writing the minutes, coaching the leaders on how to present. And that was actually led by Dr. Michael King, who's now in his retirement post-Merck period. He's a professor in the chemical engineering department here. So I got that job, lots of visibility, and it actually led to several other steps in my career where, such as this one. So when the manufacturing department needed somebody to lead all the team leaders of drugs at Merck, they thought of me. I interviewed for the job, and I got that job, which was really great because I was actually mentoring people or managing people who were doing my favorite job in the world, which was that technical leadership job. Later, while I was in that role, I was asked to come back to R&D and lead a project management department. And that was a job I had never done before, so I'd never been an R&D project manager, but they wanted me to lead a department of 30 people. So to me, that was a huge leap, and I wasn't sure I was ready to take it, but I did. And within a year, that led to what was my sort of long-term career goal. I got there much faster, which was to lead a product development team. So rather than leading the one little octagon over there, the technical team, I was leading the overall team where the technical team was just a piece of it. So the research, the clinical, marketing, biomarkers, all of that was part of the team that I was leading. And you're figuring out the strategy, you're making sure that that people are staying on track, you're adapting to changes along the way. So that's what I was doing at Merck. I also did a similar thing at Teva, and then I'll talk about how that led to the Moderna job. So next lesson really that I have is to build relationships and to maintain them. And so they can be inside the company and to me, part of it just makes life and work more fun. So this is my Takeda group. I'm the reindeer on the right. This is in Harvard Square after we ran a 5K together. It was my slowest 5K I've ever run because the little pom-poms on my reindeer costume kept hitting me in the face. But there was lots of like hard cider at the end, and it was super fun. So another thing it does is provide support when maybe you're having really big challenges and you need people to just buck you up, or provide an alternative perspective to a problem you're trying to solve. Maybe it's a new way of solving that problem. It may help you find a job. So the job that I talked about with becoming a manager of all the team leaders, the first person to tell me about it was a good friend that I met in the chemical engineering department years ago. And then once I had that job, somebody that I had mentored before turned out to be the perfect hire. So somebody with technical skills, leadership and collaboration skills. And I was able to bring her into the group, which was great. Other things that I recommend, and some of which I haven't done as well as I'd like, getting involved in scientific meetings, giving talks. Later in your career, you might be chairing sessions. My husband's done a great job with that. I feel like that's an area that I would encourage people to do, even if you think you don't have time for it, because it can be very helpful and also very fun. And also, I really loved being an advisor to the chemical engineering department. I like talking to students, faculty members, other people on the board, and that's been really rewarding for me as well. So, learning number 4, take on new challenges. So, lately I've been taking on a lot more physical challenges in addition to taking on work challenges. So that's me in the White Mountains, and then that's me in the White Mountains with my son Ben, who helped me get through it. He's getting ready to start his residency here in July. And just the one thing I also wanted to quote was a couple of years ago, there was a lecture series started in honor of Ed Paul, who recently passed away. I think that was in 2019. And he got his bachelor's back in 1952, and he gave a talk when he was in his late 80s about his time here, which was kind of amazing because it went so far back. But one of the things that he said in his talk was, don't give up because it's difficult, and it will be difficult. So sometimes you feel like you're on one of these little skinny boats like I'm on, where you're about to tip into the water, but you've just got to sort of maintain your confidence and keep going through it. And then the other picture was from the marathon I ran in October, and it was my biggest accomplishment, other than maybe some of my Moderna accomplishments last year, where I qualified for the Boston Marathon for next year. And okay, so new challenges, Moderna. I'd kind of finished up my assignment at Takeda, looking for what my next big thing was, and a recruiter called me about getting back into infectious disease development at Moderna. So I instantly wanted to do that, something I really enjoy doing, but I'd never worked at a tiny company, and at the time it was still just a few hundred people. Now it's like 3,000. We're hiring hundreds of people every week. But it's still, though, it's a lot more problem solving through science. So it still had that thread in it that I felt comfortable and felt like my background could help me contribute to. Moderna's big on their values. So you can see that a lot of these connect to what I told you about in grad school, how important it is to be curious. Collaboration's important. Being resilient is important, although it's kind of got a little harder edge at Moderna with being relentless. And I continue to try to be bold and visionary. So that to me is something I always strive to do more. So when I joined Moderna, it seemed like everybody was working on COVID. So the whole company had been redeployed to that. And they were looking to hire people to work on the other programs. And I was actually hired to work on the Zika vaccine, which is Zika is a virus that causes congenital abnormalities in children born to infected mothers. I think a lot of people heard about it back around the time of the 2016 Olympics in Brazil, because there was a big epidemic in Brazil. And it was quite serious. And that's about the time that Moderna started working on it. I also, about a month into the job, got assigned to work on a next generation COVID-19 vaccine. They were looking for something that would be stable longer in the refrigerator, so you'd have a longer shelf life. So I worked on that from July until last summer. And then much to my surprise, in June, I was asked to take over the entire COVID vaccine program on top of everything else I was doing. So before I get into sort of what I was doing, I did want to just give a shout out to all the chemists and engineers who were really responsible for what was done in 2020. So from having an amino acid sequence to FDA authorization and getting vaccine out to the public was really amazing. And the beginning of that was taking that amino acid, and this is sort of what's shown here, getting that from the sequence to actually shipping something to the clinic for clinical trials in just 42 days. And it was really built on a decade of experience of figuring out how to make mRNA vaccines, but also total focus and commitment by the engineers who are working on it. So when I came along, the state of the pandemic was that it was about 6 months after vaccines started being distributed, hundreds of millions of adults vaccinated. Vaccines for adolescents were just starting to be authorized, and the variants were emerging. So in July 2020, sorry, let's just say, both of those should say 2021. Anyway, so July last year, the Delta variant had really taken over, and we're trying to figure out what to do. Does our vaccine work against Delta? How long does it work, and so forth. So what we found out was that it worked pretty well, but over time you got waning. So you started to see breakthrough cases after several months, several meaning like 8, 9, 10, 11, 12 months. And that really led to our really collaboration with the government of determining that it was time to submit a package for a booster dose. And I was very much involved in getting all of that work together to be able to present that to the FDA in October. And then we were authorized at that time to begin the booster dose. First, older people started getting vaccinated, and then it was opened up to a broader public. So it really seemed like we'd gotten it under control. Delta was dominant. People had vaccines that worked, and then bam, the start of the Omicron wave. So we had sort of a multi-pronged approach to Omicron. First of all, we wanted to understand by taking sera that we'd had banked from our previous patients and testing them against Omicron, found out whether those antibodies that were in the sera actually neutralized Omicron. And what we found out that was okay for people who'd had 2 doses, pretty good for people who had 3. So it was looking pretty good. But at the same time, we wanted to see if we could do something better. So in parallel with doing that analysis that this paper was about, we also started creating Omicron specific, so vaccines that were specific to the Omicron version of the spike protein so that we could evaluate those in the clinic. So we got that going very fast. By January, we had material that we could start evaluating in the clinic to see if that would be better. And then after that, we layered on, well, what if we combine 50-50 mix of the original vaccine and the Omicron targeted vaccine and see if that works even better and could give you that sort of broader protection that might make it possible to withstand not only the current wave, but the next wave and so on. And we don't have any data on that yet. So that's what we're working on. But in my role, really, it's that same role with the big wheel. And my team is people from, MDs from clinical and patient safety, and people from regulatory and technical and also commercial and manufacturing. It's really like working together as a team, interfacing with our senior management, because this is our only product on the market. So we get a lot of attention from senior management to figure out what should our strategy be, how do we execute that on that strategy, and how do we pivot when we get new information. And I love what you talked about with the sort of scenario planning, because we scenario plan all the time. And how do we make sure that people are staying on track? So that's pretty much what I do, with adding in a healthy dose of interacting with the government, because the government is still funding our pediatric vaccine program. They also buy vaccines for distribution, and they're very eager to know when those vaccines are going to be ready, when we're going to get our data, when they can make a decision. And they also want to review everything that we do and sometimes try to redirect us. So, I mean, it's good. They're protecting the taxpayers. They're protecting the population. It's good. And I talk to the government 2, 3, 4 times a week, depending on which week it is, to make sure that we're staying on track. So this might be my last slide. Oh, right. So lots of problem solving through science, and a great team tackling the problems. So what you see there, I've got my pharmacovigilance person, my head of regulatory, my head of clinical, some other pharmacovigilance people, and we did get to break out and actually have a party last month, and that's what that's about. But it's a lot to solve, but we got a great team working on it. So that is my last slide, and I'm happy to answer any questions that you have.
Talk. I'm a PhD student too, so I'm currently completing my PhD in electrical engineering here. So as you just mentioned, that you started your career beginning as a engineer. So I'm kind of curious, how exactly did you try to transition from just an engineer executing on the orders of some manager into a more higher level position where you're actually planning different steps of the projects, and then so try to bring the bigger scope to the project, and then help this company to achieve better outcome.
Yeah, I mean, I think one of the things is when I moved into vaccines, and I think that it's a similar case for all sort of biologically derived products. The connection between how you make the drug and making sure that you're making it in a consistent way that works and the clinical trials is much closer than when I was making a chemical that eventually was formulated to become a drug. And so the culture in the biologic side of the business, at least this was back in the 90s, was much more collaborative with people who were involved in the biochemical engineering and manufacturing side of things, and the clinical side. And so early on, I was able to have that exposure and to sort of be on the team where all of the clinical development was happening. And that gave me, I think, the experience, or at least the view or the big picture view of what was going on, and then led to opportunities in that sense. But the other thing that I did was when I realized that sort of my weakness was not really completely understanding how clinical trials were designed and executed, taking that project management job where I got that full view of the world was really instrumental. I don't think I could have moved into a project leadership job without the view that I got in the project management role.
I have a question about hype. How do you feel about the publicly traded, the speculative sort of culture surrounding these sorts of innovations?
You mean the press release culture, sort of, or?
And yeah, the investor relations and having to roll everything up into profit and that sort of stuff.
Well, I definitely have not been in a situation where every week I have to worry about a press release and understand that. So, I mean, I think it's, at first I didn't understand it. I didn't understand why are we so focused on our sort of outward information. Can't we wait a while before we talk about this? Can't we, spend a little more time doing further analysis or wait for the next bit of data? But I think over time I've understood that it's a responsibility and transparency to the investor community, and that holding key information to ourselves isn't appropriate. So it was a real learning. I mean, at Merck we used to spend 2 months figuring out what our press release was when the phase 3 results came out, and things are just at such a faster rate. I mean, and that's kind of the type of thing that I'm getting involved in now. As I told you, the Omicron data aren't out. We know when Pfizer's going to release their Omicron data, so we need to think about what our messaging is going to be before a competitor gets out there. So it's a strange part of the job that I wasn't anticipating. I'm not going to talk about whether it's good or bad. I mean, it's just kind of, it's the way it is, and we need to adapt accordingly.
Hi. Thank you for your talk. So I'm going to ask a very weird question. So every time we hear a lot of success story from all the successful people, very less time we hear about the failure story. So is there any time in your life you felt down, difficult to overcome the situation? And if so, how did you, what was your strategy? It's just to feel motivated.
Oh yeah, I probably have a lot of them. I'm trying to think of which. Okay, so I'll tell you. So I worked at Merck for over 20 years. I got this great job. It was like my favorite job in the world, of leading project teams for vaccines. And then the new head of R&D decided that was a job that only MDs could have. That if you had that role, you needed to be the clinical lead. You needed to lead all clinical development. And so I was no longer suitable for this dream job that I had achieved in 2010. So I ended up retiring from Merck at 50 and taking a job at Teva. And fortunately, I was able to find a job where it was program leadership. It wasn't infectious disease, but I thought it was a strength for me because Teva had been a generic company, and what they were trying to do in the innovation space was very engineering-ish. They were trying to take existing drugs and how can we deliver them better with a device or a new formulation. So, I mean, that was really rough for me to lose my dream job, but I went out and tried to find a job that also used my strengths and allowed me to restart, and it worked. But yeah.
I have a question. You talked about scenario planning, and I was just wondering if you could talk about the balance, because I know sometimes I've thought about scenarios and you feel prepared because you know what could go wrong, but then sometimes you can feel overwhelmed, like going down that list of every possible thing that could go wrong. I was just wondering if you could talk about how you balance that.
Yeah, I mean, I think what you try to figure out, first of all, is, what can you actually mitigate? So that's one part. And then also understanding, trying to categorize based on, it's pretty standard, I guess, risk analysis, but it's categorizing based on probability and based on the impact of something that could go wrong. So, and focusing really on things that could be highly impactful is important. And sometimes it's too costly to mitigate all of your risks. You just run out of resources to do that. So that's another thing. Sometimes you just have to accept that that's a risk. It could have a moderate timeline impact, but we could accept it, and then you just go on.
Thank you. Hi. I understand this question might have a bit of a subjective answer, but I just started grad school over here. I'm just one semester in, and up till now I've only been used to like classes and coursework. And recently I started research, and I feel like in research in grad school, but also possibly like in the industry, progress tracks very differently from coursework. I felt like up till now, coursework, it's been so linear for me. If I study, I get a certain output, there's a fixed syllabus. But in like work and research, there's just so much that can go wrong. And like there's a lot of phases that like is also much slower. It's just not a very like consistent pace. And I was wondering if
yeah, it's like you're bringing back my past traumas.
It's just been a new change of pace for me, and I'm like, there are times where it's a lot slower than others, and sometimes it's because of like equipment failure, sometimes it's because, and I just don't know during those like slow periods how to maximize my time, I guess.
Yeah, I mean, I think that's part of where the collaboration and having other grad students that you can talk to about sort of how to get over those barriers. So whether if it's slow because you can't work on your equipment because it's broken, I mean, that's a sort of a tactical issue that maybe that's a time when you're reading papers and sort of preparing for the next thing. But if it's more that it's discouraging when you go through the failures, I mean, it's tough. And I think that's one of the challenges of grad school. I mean, there's a set amount of work that you need to do, but there's not a set amount of time. It can keep stretching. So I guess the sort of balancing the continuing forward and getting that support that you need. And then maybe sometimes if you're having an intractable problem, reaching out to others, maybe people that you wouldn't normally expect for alternative ideas, which is sort of what I was saying about my photolithography measurement cell thing. I was really completely at a dead end there, but was able to get rescued a bit with some good input.